Hello everyone, this is a study of Section 3, Regulation of Cell cycle checkpoints. A checkpoint is a stage in the eukaryotic cell cycle at which the cell examines internal and external cues and decides whether or not to move forward with division. Cell cycle progress is very critical in each cell, which operates strictly under strong surveillance to prevent mistake. So, progression of cell cycle is highly regulated in certain points that are called cell cycle checkpoints. Checkpoints halt the progress of cell cycle if any of the chromosomal DNA is damaged and DNA replication and chromosome alignment is correct and complete. Thus, checkpoints ensure various events in the cell cycle progression to occur actually in correct order. There are a number of checkpoints, but the three most important ones are the G1S transition, the G2M transition, the spindle checkpoint at the transition from metaphase to anaphase. G1 checkpoint is a restriction checkpoint which allows entry into the S phase or causes the cell to leave the cycle, entering a non-dividing G0 phase. It can be influenced by growth factors, nutrients, cell size, and DNA damage. So, it can answer the question, is the environment favorable? G2 checkpoint is the G2M DNA damage checkpoint which halts the cell cycle until specific go-ahead signals are given to continue the phase. It can be influenced by cell size, DNA damage, and DNA replication. So, it is used to check, is all DNA replicated? Is the environment favorable? AMP checkpoint is a spindle assembly checkpoint which can be influenced by chromosome attachments to spindle. The G1 checkpoint is the main decision point for a cell, that is, the primary point at which it must choose whether or not to divide. Once the cell passes the G1 checkpoint and enters the S phase, it becomes irreversibly committed to division. That is, barring unexpected problems such as DNA damage or replication errors, a cell that passes the G1 checkpoint will continue the rest of the way through the cell cycle and produce two daughter cells. So, if a cell receives a go-ahead signal at the G1 checkpoint, it will usually complete the S, G1, and M phases and divide. If the cell does not receive the go-ahead signal, it will exit the cycle, switching into a non-dividing state, G0 phase. G1 checkpoint operates at the end of G1 phase. It checks whether conditions are favorable for the cell to divide, DNA for any damage before it is going replication in S phase. If DNA damage is detected, checkpoint proteins will prevent the formation of active cyclin CTK complexes. Inhibition of cyclin CTK complexes stops progression of cell cycle. Directs the DNA repair mechanism rectifies the DNA damage. If the damage can't be repaired, the cell will go to apoptosis. G2 checkpoints include the checks for damaged DNA, unreplicated DNA, and the checks that ensure that the genome is replicated once and only once per cell cycle. If cells pass these checkpoints, they follow normal transition to the M phase. At this stage, the cell will check DNA integrity. Is any of the DNA damaged? DNA replication. Was the DNA completely copied during S phase? If errors or damage are detected, the cell will pause at the G2 checkpoint to allow for repairs. If the checkpoint mechanisms detect problems with the DNA, the cell cycle is halted and the cell attempts to either complete DNA replication or repair the damaged DNA. If the damage is irreparable, the cell may undergo apoptosis. This self-destruction mechanism ensures that damaged DNA is not passed on to daughter cells and is important in preventing cancer. M checkpoint is also known as the spindle checkpoint. a cell examines whether all the sister chromatids are correctly attached to the spindle microtubules. M checkpoint senses the integrity of the spindle apparatus in the cell, correct orientation of chromosomes in the metaphase plate, whether all chromosomes are properly bound to spindle fiber. How does this checkpoint work? It seems that cells don't actually scan the metaphase plate to confirm that all the chromosomes are there. Instead, they look for straggler chromosomes that are in the wrong place. For example, floating around in the cytoplasm. If a chromosome is misplaced, the cell will pause mitosis, allowing time for the spindle to capture the stray chromosome. M checkpoint thus prevents the incorrect sorting chromosomes. Internal and external cues trigger signaling pathways inside the cell that activate or inactivate a set of core proteins that move the cell cycle forward. CDKs, cyclins, and the APC are direct regulators of cell cycle transitions, but they aren't always in the driver's seat. Instead, they respond to the cues from inside and outside the cell. These cues influence activity of the core regulators to determine whether the cell moves forward in the cell cycle. Positive cues, like growth factors, typically increase the activity of CDKs and cyclins, while negative ones, like DNA damage, typically decrease or block activity. For example, some growth factor binds to a specific receptor protein on the plasma membrane and activates the relay proteins on the corresponding signal transduction pathway, which transfer the signal into the nucleus to affect the cell cycle control system that determines the cell cycle is going on through the checkpoints. CDK inhibitors are also involved in cell cycle checkpoints regulation by inhibition of cyclin CDK's complex activation. So, the protein's complexes delay the cell cycle progression until problems are fixed. They can prevent cell division when problems cannot be fixed and induce apoptosis if problems are so severe and cannot be repaired. In this way, the genome of the organism is maintained with only one round replication of DNA per cell cycle. Almost all cancers are due to the improper function of the proteins involved in cell cycle regulation. The RB protein is a tumor suppressor, which plays a pivotal role in the negative control of the cell cycle and in tumor progression. It has been shown that RB protein is responsible for major G1 checkpoint blocking S-phase entry and cell growth, promoting terminal differentiation by inducing both cell cycle exits and tissue-specific gene expression. The role of the RB gene family in cell cycle regulation includes the interaction between the RB family proteins and the E2F family transcription factors, which plays a central role in governing cell cycle progression and DNA replication by controlling the expression of cell cycle E2F-dependent genes. RB and RBP represents the unphosphorylated and the phosphorylated forms of the RB protein. In G0 and early G1 phases, RB physically associates with E2F factors and blocks their transactivation domain. In late G1, RBP releases E2F, allowing the expression of genes that encode products necessary for S-phase progression. The protein 53 tumor suppressor is the master regulator of divisor cellular processes. Stress input, such as DNA damage, hypoxia, replication, and translational stress, oncogene activation, viral infection, and so on, can activate protein 53 expression and cause transcriptional output, which results in cell cycle arrest, DNA repair, apoptosis, angiogenesis, and autoregulation. For example, DNA damage can and will happen in many cells of the body during a person's lifetime. Cells must be able to deal with this damage, fixing it if possible, and preventing cell division if not. Protein 53, the guardian of the genome, works on multiple levels to ensure that cells don't pass on their damaged DNA through cell division. First, it stops the cell cycle at the G1 checkpoint by triggering production of CKI proteins. The CKI proteins bind to CDK cycling complexes and block their activity, buying time for DNA repair. Protein 53's second job is to activate DNA repair enzymes. If DNA damage is not fixable, Protein 53 will play its third and final role, triggering programmed cell death so damaged DNA is not passed on. When Protein 53 is defective or missing, mutations can accumulate quickly, potentially leading to cancer. Indeed, out of all the entire human genome, Protein 53 is the single gene most often mutated in cancers. Protein 53 and cell cycle regulation are key topics of study for researchers working on new treatments for cancer. This is the end of the section. Thank you very much.